Okay. Hello, everyone. My name is Dan McManus. I'm a moderator here with Open Box Science. And today we have a fantastic back-to-back papers, two fantastic back-to-back papers in Nature Immunology on tissue-resident memory T cells. And our first speaker will be Simone Park. Simone did her undergrad work at the University of Melbourne in Australia, and she's been studying tissue-resident memory cells since she was an undergrad. Right? She did an honors thesis with Frank Carbone, one of the pioneers that founded, if you will, tissue-resident memory cells. And then after undergrad, she went to do her PhD in immunology, also at the University of Melbourne, and was co-mentored by Thomas Gebhardt and Laura Mackay, two other well-renowned T-cell immunologists. Simone stayed on then with Laura Mackay to do a postdoc, and then she went to Philadelphia, okay, to do continue her postdoc training with John Wherry at the University of Pennsylvania. Simone's very early in her career, but she's made incredible findings and contributions to our understanding of T-cell immunology and is an emerging leader. And with that, let's welcome Simone. And Simone, go ahead. You can get your slides up.
Thank you for that intro. Just a second. My computer is freaking out. Of course, we tested the slides, but now they don't want to perform. Okay, thanks for the
Looks
introduction.
beautiful.
So I'm going to talk today about a paper that we recently published on CD8 T cell differentiation in the context of chronic antigen stimulation and acute antigen stimulation in tissues. So we know that during an acute infection or after an acute stimulation like vaccination, CD8 T cells become activated and they differentiate to give rise to effector cells that will later then go on to give rise to a heterogeneous pool of memory T cells. And this pool of memory T cells includes recirculating memory T cells, such as effector memory T cells and central memory T cells that constantly migrate through the blood and secondary lymphoid organs, as well as tissue resident memory T cells that are found within peripheral non-lymphoid tissues and permanently reside within those tissues without recirculating in the blood. And it's this tissue resident memory T cell population that is really important to provide frontline protection against infection and against cancer within these barrier tissues. So at least in the setting of acute infection, these TRM cells can be distinguished from their circulating memory T cell counterparts by their expression of hallmark surface molecules that are associated with tissue retention, like CD69, CXCR6, and in certain tissues, CD103. So our TRM cells co-express many of these tissue residency associated molecules, whereas these molecules are not expressed by circulating memory T cells. And in addition, these TRM cells can be distinguished from their circulating memory T cell counterparts because they have a distinct transcriptome and epigenome. So this is what happens during an acute stimulation when everything's going well. But we also know that during chronic antigen stimulation in the setting of chronic infection or cancer, or in situations where antigen isn't completely cleared, CD8 T cells instead get diverted down a distinct or different differentiation pathway. And instead of giving rise to memory T cells, these cells will instead differentiate into exhausted T cells. And this exhausted T cell population can also be subdivided into a variety of different subsets, including progenitors that are more proliferative, have more plasticity, intermediate cells that are more effector-like, and terminally exhausted T cells that have poor effector functions and poor proliferative potential. And these terminally exhausted T cells can also be tissue resident. They're often found within non-lymphoid tissues. And it's this terminally exhausted T cell subset that shares many phenotypic similarities with TRM cells that arise after acute infection, including the expression of tissue residency associated molecules. And so because of this overlap in the phenotype of these two T cell subsets, it can be really hard to tell these two different types of cells apart in settings where we don't really have a good idea of the T cell stimulation history or the differentiation status or trajectory of CD8 T cells. And this includes in chronic disease settings like autoimmunity, but also in solid cancers, where we find a variety of CD8 T cells that upregulate molecules that are associated with tissue retention, like CD69 and CD103, as well as the TRM-associated transcription factor Hobbit. And they also co-express markers of exhaustion, like PD-1, CD39 inhibitory receptors. And it's these TRM-like TIL that are of interest in a lot of different solid cancer settings because they often positively associate with cancer protection as well as immunotherapy responses. And these cells are often annotated as TRM cells within solid cancers because they express markers that are associated with TRM, like CD69 and CD103. But we don't really understand, or at least we didn't until recently, how these cells might be related to these canonical TRM and exhausted T cell populations that have been very well described and characterized in settings of chronic infection and acute infection. And so we were interested in addressing what these cells are, how they might be related to the traditional TRM and exhausted T cell subsets that we all know and love. And so to address this question in a bit more detail, we turned to using a model of acute and chronic infection that we're very well acquainted with in the Wherry lab, and that's the LCMV model of infection. And so using this model in mice, we're able to model infection resolution or acute infection, as well as a persistent that recognizes 33 antigen expressed in both of these strains of LCMV. So we transfer these cells into mice and infect those mice with an acute strain of the virus. If we look in peripheral tissues 30 days later, then we can identify a population of tissue-resident memory T cells found within peripheral tissues like the small intestine, the kidney, and others that upregulate markers that are associated with TRM cells like CD69 and CD103, as well as CXCR6 and CD4DNA. On the other hand, if we infect mice with LCMV Clone13 infection, many groups have mostly focused on understanding the differentiation and biology of cells within the spleen, and there's been less emphasis in understanding how these cells look and what their phenotypes are within peripheral tissues. And when we look within peripheral tissues of these chronically infected mice, what we found was that we could identify populations of CD8 T cells that were expressing these TRM cell-associated molecules, CD69 and CD103, and that looked a lot like the TRM cells that were formed after acute infection. And in certain tissues, like the kidney, we found that we only found cells that were co-expressing these tissue residency-associated molecules within the chronically infected setting and not within their acutely infected counterparts, suggesting that within peripheral tissues, we identify populations of TRM-like cells or TRM cells expressing tissue residency-associated molecules within both the setting of chronic antigen stimulation and acute antigen stimulation. So we then asked whether these cells or how these cells will compare in terms of their expression of exhaustion-associated molecules. And so we focused within this population of cells that was co-expressing these tissue residency-associated molecules in either arm or Clone13 infection. And what we found was that both our TRM cells formed in the context of arm infection, shown in blue, and these chronically stimulated CD8 T cells in peripheral tissues within chronically infected mice downregulated expression of memory-associated molecules like TCF1 by comparison with circulating memory T cells, shown in gray, and upregulated molecules that were associated with terminal exhaustion like TOX. Although we note that our chronically stimulated T cells had higher expression of TOX compared to TRM cells still displayed an upregulation of TOX by comparison to circulating memory T cells. And we found that this upregulation of an exhaustion-associated phenotype extended also to their expression of a variety of different inhibitory receptors like CD39, PD1, TIM3, etc. So both Armstrong-derived TRM cells and our chronically stimulated tissue-localized CD8 T cells formed after Clone13 infection appeared to upregulate similar residency and exhaustion profiles. So we wondered whether, however, we noted that the chronically stimulated cells did have a higher expression of exhaustion-associated molecules. So then we wondered whether this would translate into a difference in their exhaustion profile or whether this would translate into a functional difference between these subsets. And so to test this, we asked whether these cells had a difference in their ability to respond to peptide stimulation. And we found that indeed the cells derived from chronic infection had reduced capacity to produce interferon gamma, TNF effector cytokines by comparison with TRM cells from Armstrong infection. And also these cells had a reduced ability to proliferate and differentiate when they had been re-stimulated with Armstrong rechallenge, suggesting that these cells were both less functional and less plastic. And so what we found was that within chronically infected tissues, we noticed that there was a subset of cells that looked a lot like TRM cells, but had reduced functionality and reduced plasticity. But it was unclear to us from these experiments whether these cells were simply a subset of TRM cells that was more dysfunctional or a dysfunctional subclass of TRM, or whether these cells constituted a separate T cell lineage with potentially different molecular regulation and developmental potential. And so to address that question in more detail, we turned to doing some molecular profiling and looking at the transcriptional epigenetic programs of these different T cell populations. And so we used T sequencing, which is a technique that allowed us to profile 3 different modalities within the same single cells. So protein expression, as well as epigenetic landscapes or ATAC, and transcriptional information. We transferred P14 cells into mice, infected mice with LCMV Armstrong and clone 13, and then isolated cells from a variety of different tissues and subjected those cells to T sequencing. And using this technique, we were able to resolve or identify cells from the small intestine, the liver, and the salivary gland and the spleen from both acutely and chronically infected mice and compare their gene expression. So when we compared gene expression between TRM cells derived from Armstrong infection and chronically stimulated T cells derived from clone 13 infection from the same tissue, we found that those cells both upregulated expression of genes that were typically associated with TRM cells, things like CD103, RUNX3, BLIMP1, and in certain tissues also Hobbit or ZFP683. As well as upregulating expression of markers or genes that were associated with terminal exhaustion, suggesting that there is a lot of overlap in the gene expression between these 2 different T cell populations coming from the 2 different infections. However, when we look at the global transcriptional and epigenetic profiles of these cells, we found that although our Armstrong TRM cells all clustered together within transcriptional epigenetic PCA space, the cells derived from clone 13 infected tissues clustered away from these Armstrong TRM cells and instead clustered more closely with terminally exhausted T cells derived from the spleen. And so we dubbed these cells tissue-resident exhausted T cells because they were epigenetically and transcriptionally distinct from TRM cells, and they had a more similar epigenome and transcriptome to terminally exhausted T cells derived from the spleen. And so this suggested to us that these cells likely had distinct underlying molecular regulation. So we wondered whether TRM cells and these tissue-resident exhausted cells would therefore have unique developmental requirements. And we first looked at tox because we know that tox is really important for the development of exhausted T cells. But we also know that TRM cells, I showed you before, do express tox at higher levels than circulating memory T cells. However, the role of tox in TRM cell development was unknown. So we asked whether tissue-resident exhausted T cells and TRM cells would have a difference in their dependencies on tox. And to do that, we used a CRISPR approach where we deleted tox or CD19 as a control within P14 cells that were congenically marked. And we co-transferred cells that had been perturbed for either tox or CD19 into mice that were then infected with Armstrong or clone 13 and looked 30 days later by flow cytometry to ask what the impact of deleting tox using CRISPR would be on the development of cells within those tissues in either context. And so when we look within the spleen, as we would expect based on what's been published, deleting tox has very little impact on the ability of memory CD8 T cells to develop within the spleen, and we don't see a defect in memory CD8 T cell development when tox has been deleted, whereas our exhausted T cells formed in the context of chronic infection are highly tox dependent. So deleting tox has a massive impact on the ability of those cells to develop and persist within the spleen. When we looked in peripheral tissues, we found a very similar trend where our tissue-resident exhausted T cells were very highly dependent on tox for their development and survival, whereas TRM cells, although they were expressing tox at a protein and a gene expression level, they did not require tox for development or survival, and deleting tox had very little impact on their numbers or their function. And so we found that therefore tissue-resident exhausted cells required tox for both development and survival, as well as for the induction of their expression of residency molecules. I'm not showing that data here, but it is in the paper if you want to see it. Whereas although TRM express tox, they don't depend on tox for their survival or for their induction of residency molecules or residency programming. So this showed to us that TRM and tissue-resident exhausted cells depend on different molecular regulators for their development. But we wanted to know if this means that these cells are distinct cellular lineages. And so to begin to get at that question, we asked whether these cells could interconvert and give rise to each other. So we first asked whether TRM cells would become exhausted if they could become exhausted if they were exposed to chronic antigen stimulation. And so to do this, we generated populations of P14 cells that were TRM cells within the small intestine or the liver, as well as circulating memory T cells in the spleen. And then we sorted those cells out of Alcine B. Armstrong infected mice and retransferred those cells into new mice that we then infected with clone 13 to provide a source of chronic antigen stimulation. We asked 21 days later if those TRM cells that had been transferred had become exhausted. And what we found was that those TRM cells that had been re-stimulated with clone 13 had upregulated expression of inhibitory receptors and TOX to much higher levels than in situ TRM cells that had not been re-stimulated, shown here in the purple, as well as TRM cells that had been re-stimulated with Armstrong rechallenge, as well as having reduced ability to produce cytokines, such as interferon gamma and TNF, suggesting that these cells had become functionally exhausted. And again, I'm not going to show this data here today due to time, but we found that these TRM cells were capable of giving rise to all of the different exhausted T cell subsets, including the most plastic exhausted progenitors, although they had a bias towards giving rise to more terminally differentiated cells. So this indicated to us that TRM can become exhausted when they're exposed to chronic antigen. What about tissue-resident exhausted cells and exhausted cells in general? Can they give rise to TRM? So we performed basically the reciprocal experiment where we generated populations of exhausted T cells by infecting mice with clone 13 progenitors, intermediate, such that the cells would be re-stimulated, but antigen would be cleared within around a week, and these cells would then be experiencing antigen withdrawal. And then we asked whether these established exhausted T cells would become TRM cells following antigen clearance. And so, as expected, we recovered few intermediate or terminally exhausted T cells following retransfer and rechallenge with Alcine B. Armstrong. However, we did recover a larger population of exhausted progenitor cells, and these cells were detectable within peripheral tissues as well as in the spleen. However, when we looked at the phenotype of those cells and we compared them to naive T cells that had been stimulated with Alcine B. Armstrong, we found that they had a very different phenotype. So those cells were not able to efficiently acquire a residency program. They had trouble upregulating molecules that were associated with permanent tissue residency, and they seemed to retain more hallmarks of recirculation, suggesting that cells derived from exhausted progenitors are incapable of forming TRM cells following antigen clearance. But we know that within chronically infected mice, we do see a population of cells that upregulates these tissue residency associated molecules within peripheral tissues. And so where are these cells coming from? We wondered whether progenitors would be capable of giving rise to cells that had these tissue residency associated molecules when they were re-stimulated with chronic antigen. And so we therefore performed the same experiment, but we instead transferred these populations of exhausted T cells into chronically infected mice and asked whether these cells would then give rise to tissue-resident exhausted cells. And what we found was that in this setting, these exhausted progenitors were capable of upregulating tissue residency associated molecules. And so this suggested to us that these TRM cells and tissue resident exhausted cells are separate developmental trajectories or separate lineage differentiation pathways, and that they derive through separate or distinct differentiation trajectories. Okay. So now, given that we'd shown that these are two different subsets of cells, two different cell states that form via distinct developmental pathways, we wondered which of these cells was the type that was associating with improved disease outcomes and improved responses to checkpoint blockade, like I mentioned at the beginning of the talk. So these populations of cells found within tumors that co-express molecules of exhaustion and molecules of tissue residency could be either of these flavors. They could be TRM, they could be tissue resident exhausted T cells. And using these markers that many people have been using to sort of try and deconvolute these cells within the tumor microenvironment, it wouldn't really be possible to tell the difference. That's because they have shared surface molecules and they have very similar gene expression. So I showed you that they have this overlap and exhausted T cells by comparison with circulating memory T cells. And for that reason, established signatures that people generally historically use to identify or to try and deconvolute TRM cells within disease settings, as well as terminally exhausted T cells within disease settings, these signatures do not perfectly deconvolute these populations within our data set. So the TRM core signature that's previously been established is enriched both within bona fide TRM cells from Armstrong infection, as well as tissue-resident exhausted T cells found within these peripheral tissues. And terminally exhausted T cell signature is enriched not only within tissue-resident exhausted T cells and exhausted T cell populations, but also to some degree within TRM cells compared to circulating memory T cells. So we needed to derive potentially better signatures that would allow us to deconvolute these subsets. And given that we had generated our own data sets comparing these cells within peripheral tissues, we wondered whether we would be able to do that. And so by comparing the gene expression between TRM cells and tissue-resident exhausted cells within the peripheral tissues we'd analyzed, so the small intestine, liver, and salivary gland, we were able to derive new signatures that more exclusively were enriched within the TRM cell population without being enriched in tissue-resident exhausted T cells, or more specific to the exhausted T cell lineage. And using these signatures, we were then able to show enrichment only within TRM cells for the TRM-specific signature, or a greater enrichment within the tissue-resident exhausted population for the tissue-resident exhausted specific signature. We were also able to identify a variety of surface molecules that were enriched either in tissue-resident exhausted cells or within TRM cells. And this included two molecules that we focused in on, which we noted have been either upregulated in tissue-resident exhausted T cells, here CD200R, or upregulated in TRM cells, here CD73, compared to the opposing subset. So if we look within cells in chronically infected tissues or after Armstrong infection that express equivalent levels of CD69 and CD103, we find that TRM cells have high expression of CD73 and low expression of CD200R, whereas cells derived from chronic infection have the inverse phenotype. Suggesting that these markers might be able to better deconvolute these subsets. And when we look within human samples, human tumor and healthy tissue samples, we could identify populations of cells that had expression of CD73 and looked more like TRM cells within healthy tissue sites, whereas cells had higher expression of CD200R and looked more like tissue-resident exhausted cells within tumors or tumor samples. So this suggested that maybe these signatures and markers might be able to better deconvolute these subsets. And then we could ask the question of which of these subsets is more associated with disease protection and also responses to immunotherapy. So we took some public immunotherapy patient databases and we took our different signatures that we had generated and we asked whether there would be a difference in the association of TRM cells or tissue-resident exhausted T cells with patient outcomes. And we first looked at a data set where patients had been treated with anti-PD-L1 in this urothelial cell carcinoma data set, where it had previously been shown that there was a relationship between CD103 and patient survival. And so they had attributed this as potentially being related to the development of TRM cells, but obviously CD103 could be expressed by either TRM cells or tissue-resident exhausted T cells. So using our new signatures, we were able to show that it was the tissue-resident exhausted signature that seems to associate with improved survival within this cohort, whereas the TRM-specific signature wasn't really associated with any patient benefit, suggesting that tissue-resident exhausted T cells might be the subset that's more associated with improved responses to checkpoint blockade. And we saw similar things within a PD-1 CTLA-4 immunotherapy melanoma cohort as well. But we wanted to functionally test, therefore, whether this indicates that there might be a potential difference in the ability of tissue-resident exhausted cells and TRM cells to respond to immune checkpoint blockade. And for that, we turned back to using our mouse model, our mouse studies, and we did these experiments where we asked whether TRM and tissue-resident exhausted cells could directly have a difference in their ability to respond to PD-1 blockade in vivo. So we transferred P14 cells into mice that were infected with LCMV Armstrong or clone 13. And then 4 weeks later, we treated these mice with FTY720 to block T cell recirculation or infiltration into the tissues and isolate the effects of PD-1 blockade on cells that were found within the tissue. We then treated mice with anti-PD-L1 or with a vehicle, and 48 hours later, so a very short time point, only 2 days, we harvested the tissues from these mice, and we asked whether the cells found within the tissues had a different ability to produce cytokines following peptide restimulation. And what we found was that for TRM cells, adding PD-L1 did very little to improve the function of those cells and didn't really boost their ability to produce TNF and interferon gamma, whereas our tissue-resident exhausted cells did experience an increase in their ability to produce effector cytokines following PD-1 blockade. And so this is just summarized here, where you can see that TRM cells do not really seem to be responding to anti-PD-L1 treatment, whereas our tissue-resident exhausted cell population has an intrinsic ability to respond to PD-L1 blockade. And so this suggests to us that these CD8 T cells that thrive under different circumstances have distinct developmental trajectories and distinct differentiation pathways, and that this has consequences for the functions of these cells and also their responses to therapy. So both TRM and tissue-resident exhausted T cells infiltrate tissues and upregulate a residency program, but they have distinct abilities to, they have distinct developmental requirements, so they depend on different transcription factors, and they have distinct abilities to respond to different therapeutic interventions. So whereas TRM cells, when restimulated with antigen, have a heightened ability to proliferate, a heightened plasticity potential, these cells can give rise to other types of CD8 T cell states that might therefore then be able to contribute to disease control. These cells might be therefore better suited towards immunotherapeutic interventions like cancer vaccination or cell therapy, where these therapies depend on the plasticity potential of the TRM cells themselves. On the other hand, we found that tissue-resident exhausted cells seem to be more susceptible or more modulated by immune checkpoint blockade, whereas TRM cells don't really seem to be modulated by immune checkpoint blockade. So this suggests that these distinct different therapeutic interventions are targeting different populations. And therefore, we need to consider the types of T cells that we have within disease settings before we can decide how we might best treat patients and what sorts of therapies or interventions might be the best choices to move forward strategies. So I need to think about when these cells might be generated in different contexts. And the way that we're thinking about this is that when we have complete antigen clearance, such as in the setting of viral clearance or vaccination, we get obviously TRM cell formation. We might also see TRM cell formation in certain settings where you have antigen persistence, but the antigen burden is very low, such that CD8 T cells are only being re-stimulated intermittently, or they're experiencing breaks in their stimulation, or the antigen is being compartmentalized in some way. On the other hand, when we have a high antigen burden or we have total antigen persistence, such as in settings like chronic infection, progressively growing tumors, but also potentially autoimmunity or transplantation, cells might be more inclined to differentiate into tissue-resident exhausted cells. And because these cells have different molecular regulation, different developmental requirements, and a different capacity to respond to therapeutic intervention, we need to think about what types of cells we're likely to be generating in these different contexts if we're going to devise better treatments for these different conditions. So I'll just thank everybody that was involved in this study, especially John and the rest of the Wherry lab for all of their help, especially in doing the large sequencing experiments that were required for this study. And then also all of our collaborators at Penn that gave us human samples and have helped contribute to this project. And happy to take any questions. Thanks.
That was great, Simone. Congratulations on the lovely work. While we wait for some other questions to come in here, I can start. I'm curious about your tox experiment. I was wondering, Simone, if you did a version where you sorted out first your T-exhausted resident memory cells, and then knocked tox out. I'm only asking because I'm curious if it's them that requires tox versus, say, the cell that gives rise to them that maybe requires tox.
Yeah. So that's a good question. So we didn't do it in the way that you're describing, but we do have like a conditional tox knockout mouse. So we did do those experiments, and like it's kind of similar to what happens in the setting of chronic infection when you delete tox. Like you do have a decline of cells, but it's kind of like hard to tell exactly what population you're targeting when you delete tox, right? So like it could be that you're removing progenitors and then stopping the formation of the cells that then give rise to tissue resident exhausted cells. I don't really know if tox is required for the sustained like programming of tissue resident exhausted cells over time. That's something that it would be interesting to look at in the future. We haven't really had the ability to do that. The problem is if we sort out the tissue resident exhausted cells, there's no real way to put them back in, right? So we need like a conditional deletion system where we could delete tox just within that population and not the progenitor population in vivo. And we don't really have a good way to do that yet. But maybe in the future, hopefully, with some of the techniques we're trying to develop, we can address that question.
Very cool. Awesome. Yeah, future papers. So we have Wassihan Hale Michael. Do you want to, has his hand raised? Would you like to ask the question?
Yeah, thank you. And congratulations, Simone, on the paper. Seems very interesting. I just want to ask, I don't have a lot of experience with mice, but when you knock out tox, do you think the mice will survive the tox knockout? Because I thought they might have more immunopathology, or was there any conditions that happens after tox knockout?
So in this particular setting, we're just knocking out tox from the P14 cells that we then transfer in. So the mouse itself does still have tox. So it won't impact like other things that are happening when we're knocking out tox in these specific experiments. Does that answer your question?
Yeah. If it's okay, I do have another one actually.
Yeah.
Yeah. So this tissue-resident exhausted T cells, do they also have transcription factor that maybe pops up with single-cell data or with ataxic, which is maybe distinct, that potentially would be like master regulator or transcription factor for this population if its tox is not programming them their potential?
So tox is programming the tissue resident exhausted cells. So it is required for tissue resident exhausted cells. But like in answer to your question, yes, there are certain transcription factors or molecules that are enriched within the tissue resident exhausted cell population compared to TRM cells and also compared to splenic cells. However, like it seems that the vast majority of the tissue resident exhausted program, tissue resident exhausted T cell program, I should say, is kind of like a combination of the exhaustion program overlaid with the tissue residency program. TRM cells, on the other hand, kind of have their own unique set of genes that are only found within TRM cells, whereas tissue resident exhausted cells seem to be like an additive combination of exhaustion plus shared tissue residency genes. And by that, I mean genes that they share with TRM. However, there are like some things that are enriched more within the tissue resident exhausted cell population, just not things that I would describe as like cell state specific compared to other exhausted T cell subsets. Compared to TRM cells, yes, definitely there are things that are exclusively expressed in the tissue resident exhausted T cell subset, but compared to other types of exhausted T cells, including splenic terminally exhausted T cells, that is kind of like more of a gradiated expression or increase in expression rather than exclusive expression compared to those cells. Does that answer your question?
Yeah, sure. Thank you. Congratulations once again.
Thank you.
And then just very quickly here, Simone, we have a question in the chat from Sylvester Maroney. They say, hi, Dr. Park, have you applied your signatures to human PBMCs to see if you can identify T-exhausted versus bona fide TRM? Thanks so much.
There's a small supplemental panel in the paper if you're interested that applies the signatures to human PBMCs. And that's a data set that comes from a previous paper from our lab. In that data set, it was like a bulk sequencing, so we can't really resolve like XTRM, which is I presume what you're talking about when we're talking about PBMCs. In answer to your question, we do see that we see an enrichment for the exhaustion signature a little bit within circulating exhausted T cells, but not the TRM signature. But in terms of whether the TRM signature could enrich for XTRM that are found in the blood, I'm not super sure. We haven't done that analysis.
Okay, awesome. Thank you, Simone. All right. Well,
No worries.
Tom, would you like to get...
sharing.
Yeah. All right. All right. So our next speaker is Tom Byrne. Tom also has an incredible pedigree in terms of T-cell immunology and immunology in general. So Tom did his undergrad at the University of Auckland in New Zealand. He then did a PhD in immunology at the University of Pennsylvania in the US under the guidance of Dr. Edward Behrens. Then Tom went back to or went to Australia, excuse me, to do a postdoc with Laura Mackay at the University of Melbourne. So yeah, Tom, you and Simone just swap places, it seems. Yeah.
We
If
did.
you'd like,
Yes, we briefly overlapped.
yes, okay, awesome. Go ahead and share your PowerPoint there.
Great. Thank you, Dan. Thanks for the introduction and thank you for inviting me. Yeah, it's great to be able to present. Oops, what happened? There we go. Just move. Okay, so present our recent study. So I'm not going to belabor this point because Simone's done a great job of outlining the development of these cells, but just to set the scene for how we came at this question, I will give a brief introduction. So it's well appreciated now that our T cell memory populations can be roughly divided into those that circulate and those that reside in our tissues long term, known as tissue resident memory T cells. And this residency has been demonstrated by experimentally by tissue transplantation and parabiosis studies. And it's revealed that the majority of CD8 T cells that are found in tissues are actually resident and they're in disequilibrium from those in the blood. TRM and circulating T cells are phenotypically distinct. And so, you've heard already today, markers including CD69 and CD103 are used frequently to identify resident memory T cells. And across organs, resident memory T cells share a common gene signature. And at the base of this gene signature is a program to essentially shut down migration so that they do no longer move. And over the past few years, cells with a tissue resident memory phenotype have been associated with positive prognoses in cancer. So it's been a very hot topic of study, understanding how these resident memory T cells are eliciting anti-cancer immunity. So resident memory T cells and T cell are actually difficult to prove. So in terms of what resident means, a non-migratory T cell, we can't actually often prove that cells are resident in experimentally in humans, aside from some transplantation studies showing that, T cells present from the donor in the transplant can persist very long term. We don't often have experimental proof of residency. And the other concept of what T cell memory is, in our kind of view, this is something that exists, or T cells that exist post the clearance of antigen. And the antigen specificity of T cells and the availability of antigen within tissues coming from humans is often unknown. So to kind of understand or find resident memory T cells in humans, people have relied on, cell surface markers or transcriptional profiles to discriminate them from circulating memory cells. And so, us and others have developed TRM gene signatures, basically comparing the signatures between resident memory and circulating memory T cells. However, this becomes more complex in settings where antigen may persist and T cells get driven down this alternative differentiation pathway towards T cell exhaustion. And T cell exhaustion signatures have essentially been created similarly, where it's with differentially expressed genes between exhausted T cells and circulating memory T cells. And the problem with this is that the resident memory and the exhausted T cells have not been, specifically compared. And actually, the exhausted T cells, as you've just seen this morning from Simone, have basically express the core gene signature. And so this is just a point to show how like overlapping this is. If you look at where the core TRM gene signature is enriched within T cells from chronically infected mice, it's exactly the same T cells that, T cell exhaustion people would identify as exhausted or terminally exhausted T cells. So terminally exhausted T cells adopt a residency signature. And the problem when we go and use these signatures in the settings of cancer is that if you overlay a TRM signature on T cells coming from cancers, even if there are both TRM and exhausted cells there, these signatures will see all of the cells as resident memory T cells. And experimental evidence has shown that exhausted T cells do actually become resident in tumors. And so this is data from Dave Mazepis' lab a couple of years ago, showing that the more terminally exhausted T cells are the most resident within tumors in parabiosis settings. And so we thought that maybe the known functions of exhausted T cells in tumors may have been incorrectly ascribed to resident memory T cells based on this shared signature expression. So what we wanted to do was, see if we could deconvolute these populations. And, the problem is what we expect is that using these canonical protein markers for TRM, 69 and 103 on CD8 T cells isolated from tumors, we'd expect that this gate likely contains both resident memory and exhausted T cells. And this is from human breast tissue. So we wanted to determine whether, both of these populations do exist within tumors and whether they are distinct cell types. So to do this, we made two assumptions. The first being that same phenotype cells from tumors are a combination of both exhausted and resident memory T cells. And so to do this, we performed site-seq on CD3 positive T cells from tumors and associated healthy tissue. In this case, breast cancer tumors and cancer-free breast tissue. We identified a number of populations of resident memory T cells or resident phenotype T cells expressing CD103, CD69, another, the other, conventional residency marker. Hobbit, the transcription factor, that is important for TRM formation, and downregulation of KLF2, which is important for recirculation. So, coming back to our assumptions that we made, we then looked at where these specific cell populations were isolated from. And you could see that two of these TRA or resident phenotype populations predominated the cells that came from the healthy tissue. And so we annotated these as resident memory cells, while there are select populations that were only present in the tumor-derived tissue, which we then annotate as the exhausted T cells. So, Simone talked about this just before as well, talking about how, exhausted T cells adopt this resident memory kind of signature, core signature, to, maintain their residency as well in tissues. And so if we overlay, previously published resident memory gene signatures on these populations, they do not split the TRM and exhausted cell populations in our data, whereas the exhausted cells have this added layer of a T cell exhaustion signature as well. So we generated specific signatures that could deconvolute these two cell types. And this is circular, but just showing that these signatures do highlight the cells of interest. But because we also had site-seq in here, we could also identify cell surface proteins that could be used for more functional analyses to segregate these cells. And we have consistent markers with Simone across mouse and human, such as CD73, that specifically identify TRM within that CD103, CD69 population, and then checkpoint molecules and other markers of exhaustion that are specific for exhausted T cells. So we wanted to see how kind of broadly applicable our signatures may be. So we used or turned to the Pan-Cancer Atlas that was published by Zemin Zhang's lab in 23. And we could show that if we overlay our exhausted T cell signature on T cells isolated from many different cancer types, this highlighted cells that were only identified in the tumor and not really in the healthy tissue. Whereas you could see that TRM cells enriched for the TRM signature were identified in both healthy tissue and tumor. So these signatures are able to discriminate these populations across tumor types. Okay, so this is great. But what we really wanted to do was look at some of the key things that might differ between these cell populations. So that includes functionality, the antigens that they may be specific for, as well as the developmental pathways. So the first thing that we were able to show is using, these newly identified cell surface proteins that do discriminate these populations, that the resident memory cells were more functional than the exhausted T cells. And, we had these definitions that I proposed earlier. There's a third definition here, and that's exhaustion. So that means functional impairment. And this is actually something that we can or were able to show. So does any of this matter? And this is a question that we, I guess, threw around a lot while we were going through this project, because we, didn't want to just be, stuck in semantics of, naming cell types. We don't want to do this thing of putting cells in boxes and for the sake of it. And especially what we don't want to do is gatekeep certain boxes and say, this is not a TRM, don't put it in my box. This is something that we were conscious of not doing. So what we did want to do was determine whether TRM and exhausted T cells might have different roles in tumor control. And so we looked at, because we were focused initially on breast cancer, we looked in breast cancer data. And or elevated expressive TRM signature had a good overall or better overall survival long term, and this was an inverse for those that had a high expression of the exhausted cell signature. However, and consistent with what Simone just showed, the patients that had higher expression of the exhausted T cell signature were better able to respond to checkpoint blockade, whereas those with a high expression of the TRM signature did not have any improved responses to checkpoint blockade. So the TRM did not appear to associate with checkpoint blockade responses, and so why might this be? So one thing we were able to do is across a number of different data sets, look at the relative ratio of exhausted T cells to TRM in various different cancers. And, without focusing on all of this, I just want to point out that in melanoma, consistently there were an elevated number of exhausted T cells to resident memory T cells. And one thing we know about melanoma is it has a relatively high mutational burden. And so actually when we look at the relative ratio of exhausted to TRM across these cancers, we could correlate this with tumors that typically have a high tumor mutational burden. So this predicts that exhausted T cells and TRM have distinct TCR specificities, and that exhausted T cells are tumor antigen specific. So does this mean then, in converse, that tumor-associated TRM are not tumor-specific? So because we can't organs, we turn to mice, and so we transferred either tumor-specific or bystander T cells into mice bearing B16 melanomas, and this was performed by Mackay Osman, actually, Simone's PhD student. We then performed single-cell RNA-seq on those tumors. We could show that the bystander and tumor-specific cells clustered distinctly, and that when we overlay our new human TRM and exhausted cell signatures onto these data, that the TRM signature highlights cells that are not specific for the tumor, while the exhausted signature is enriched in those that are specific for the signature for the tumor. So yeah, the breast cancer TRM signature appears to be associated with bystander TILs. And this is consistent in a different tumor model. So this is a breast cancer tumor model, same setup. We transfer an antigen specific or bystander cells, and the bystander cells are more efficient at adopting a resident memory phenotype. And this extends to those tumor-specific cells that have a low affinity for the tumor antigen. So in this case, both the OT1 and OT3 are specific for OVA, but at high or low affinities. So this would predict that in our human data sets that the TRM and exhausted T cells may be clonally distinct. And this is exactly what we see. So, across both our breast cancer data set and the pan cancer atlas from Zimin Zhang's lab, TRM and exhausted T cells appear to have distinct clonalities. And so virus-specific T cells have been identified in tumors before. Studies from including Evan Newell's lab have shown this. And Paul Thomas's group has a great data. When we identify virus-specific cells, they very frequently have a TRM phenotype within the tumor, and very rarely, if ever, have an exhausted T cell phenotype, which you may expect. So this data suggests that tumor-associated TRM may be tumor-independent bystanders or tumor-specific cells that are currently not receiving antigen-specific stimulation. But what we're not saying here is that all TRM are bystanders. And in fact, of the cells in our data sets, or in our breast cancer data set, as well as the pan cancer atlas, very few of the cells have a predicted virus specificity. So we can't say anything about the majority of the cells. And even in the mice, not all the tumor-specific T cells become exhausted. And some of these tumor-specific cells, 20% maybe, adopt this resident memory phenotype. And this actually correlates with the clearance of their antigen. So we can measure antigen through the GFP expression on the tumor cells. And you can see that as the antigen is lost, the TRM, the frequency of antigen-specific TRM is increased. And this is converse to the more terminally exhausted populations that are, correlated with high levels of tumor antigen. So one thing we also wanted to determine was whether tumor-specific TRM and exhausted T cells might be distinct T cell lineages. So to do this, we used the splinter barcoding system that was developed by Mark Dawson across the road here in Melbourne at the Peter MacCallum Cancer Centre, where we barcoded, tumor-specific T cells, transferred them into mice with the breast 83 over breast cancer, and then sequenced the barcodes from these cells isolated from different populations. What we are able to see is that is not a distinct progenitor that seems to give rise to the exhausted or resident memory populations. And so they, at least from the progenitor stage or from the naive T cell stage, naive T cells can give rise to both. But what is interesting is that the circulating and tumor infiltrating T cells are distinct lineages, or largely distinct. And this is something we'd really love to follow up, determining whether this is just a stochastic event where some T cells enter the tumor, then they adopt all these phenotypes, whereas some don't ever enter the tumor, versus this being a pre-programmed uh, um, program that is that would be very exciting. So in summary, TRM and exhausted T cells coexist in tumors and tissues. The core TRM gene signatures are adopted by exhausted T cells and cannot discriminate these two populations in tumors. Bystander T cells can enter and adopt a resident memory fate, and are currently untargeted by current checkpoint blockade therapies, which I think is an exciting area, because lots of tumors have lots of TRM that may not be, or are underutilized. Tumor-specific TRM develop in conjunction with antigen clearance, and TIL populations are a distinct lineage from circulating tumor-specific cells. So we had, and I've just got one more slide after this, just to show, just to kind of think about why we get this response of how, the TRM signature doesn't, or predicts good long-term survival. And so one hypothesis is that, TRM formation is purely coincidental with tumor regression. You get a good immune response, you clear tumor antigen, you're more likely to maintain, your, a cancer free progression long term. Something we're really interested in looking into is whether TRM provide bystander mediated functions, so, as other cell types like NK cells would. And consistent kind of with Simone's PhD work from Thomas Gebhardt, and also with Laura, showing that these TRM cells exist to maintain a tumor immune equilibrium, so that they are ready to respond, or like just keeping the tumor cells intact. So if this is the case, what we'd predict is that tumor-specific TRM might be identified in surrounding tissue. And so what we did was we isolated, we identified shared clones from a data set from Jane Grogan's lab from both tumor and associated healthy tissue. We annotated the cell types by tissue using our signatures now. And what we're able to show is that tumor-derived clones that are identified as exhausted T cells in tumors are this number of different cancers. And this kind of supports the idea that these cells might protect long term once these tumors are resected from the patients. Supporting this long term tumor control. And with that, I'd like to acknowledge everyone who kind of contributed, and this was a lot of people, and owe a lot of thanks, especially to Laura, who's been an excellent mentor to me. Malika, Philippa, and Ali, who did heaps in this paper. Mark Dawson's lab, the computational sciences people at University of Melbourne, and others, including discussions with John and Simone over the years. So with that, I'd like to take any questions.
That was fantastic, Tom. Beautiful talk, and congratulations again on the awesome paper. I'll start with a quick question. It's a bit of a philosophical one, so don't mind me. And that
Sure.
is, and perhaps Simone too can answer it at the same time. But, is it worthwhile doing away with using the term residency as a cell classifier or identifier, and maybe treat it more so it's a function, a function of a cell. Multiple cells, of course, can become resident to a tissue. What are your thoughts on that?
Yeah, I think that's spot on. I think residency is a feature of a cell, that a resident memory cell is resident, an exhausted T cell becomes resident. Um, I guess this kind of goes to the point I was making about, how much of this is semantics. I do think that there is value in, you know, being able to identify these cells distinctly. And I think, both Simone's paper and ours showed that, there is potentially an untapped resident memory population that is, targetable with future immunotherapeutic approaches. Um, yeah, so definitely residency is a feature, um, not a cell type, is if that's yeah, if
Yeah,
that's
beautifully
answering the
said.
question.
Yeah, very nicely said.
Yeah.
And while we wait here, if anyone has questions, by the way, raise your hand or type it into the chat. It's Simone, did you want to address that too? Not to.
Oh, I can. No, I agree with what Tom is saying. Great talk, by the way, Tom.
Mm.
I think like, yeah, definitely residency isn't like a property that can be acquired by a cell. That said, though, I think that there are unique features of TRM cells as like a cell state. Those cells are resident, those cells are memory cells, but they are kind of like a subset of cell that has unique features that are not related necessarily to residency. And so I think that there's still value in defining what a TRM cell is, but maybe just not in the way that people are thinking about it as it being like the residency is the most important feature of a TRM. I think there are like other properties of TRM that we can potentially hone in on or capitalize on, by virtue of the fact that they have a sort of unique regulation in comparison to circulating cells, memory cells, I mean, and exhausted cells that isn't so much related to the fact that they are resident. Does that answer your question, Dan? Hopefully. But yeah, I think
Fantastic.
residency in itself is a state.
Fantastic. Well, I would look forward to you guys writing a review together on it.
We should.
Yeah. Fantastic. Okay. All right. Let's see. All right. It doesn't look like any more questions here. So I guess, oh, here we go. Eugenio has one. He's asking, do we know the niche that maintains TRM in tumors? Any signals like cytokines like TGF-beta?
It's a good question. We were able to show that so we actually did kind of address the TGF-beta question in the paper, where we knocked out the receptor, and showed that these bystander, TRM were unable to form. So yes, I think TGF-beta is important. Um, the specific niches, and whether they're distinct, between the resident memory and the exhausted cells in the tumors, I think we need to understand more. It's something we're really interested in looking at. We did show that, through imaging, that both populations are present in, pretty similar locations. But I wouldn't say we had the granularity to go and look at, really detailed niches and what the other cell types that are interacting with that were. And I think that's a super important question, considering, lots of people now have shown the niches that are required for exhausted T cells, or function and of and sorry responsiveness to checkpoint blockade and things like the presence of TLS and tumors, and yeah, those things are important, and I don't think it's been addressed for resident memory cells yet. But good question.
Okay. Awesome. All right. Well, with that, I think we will conclude. Want to thank Tom and Simone again for coming on and presenting their awesome papers. And for anyone that's still there, next week, join in Thursday, 11 a.m. Eastern. We have another immunology seminar on myeloid cells, aging, and TGF-beta signaling. So it's a perfect intro for that seminar. Yeah, all right. Anyway, thank you guys.
Great. Thank you.
Thank you.
Thank you.